Good morning, all. It's always fun to speak at RNA Biology Conference. And I'm just thinking when I've been so erratic in attending the RNA Biology meetings, I'm just thinking, when did I speak last? My students do go. They went to the one in Delhi. So I go between RNA Biology and yeast biology. I managed to cover once in 2 years or so. But the last time I spoke, was just before the pandemic when Christine decided that she was retiring, Christine Guthrie at UCSF. So Christine was not my official PhD mentor, but we had a joint collaboration with John Abelson. So I spoke there and I met really the big granddaddies. And I was the only PhD student who was invited from Christine and John's joint work. So it's like your first love, your first kid, your first this. So all of my PhD work is on RNA biology, but always with the lens of genetics. And many of the factors that I'm going to speak on three, mostly on two factors, just two of them. Are those mutants in those I discovered, amongst 15 other factors which came out of the genetic screen. But it's just amazing how the wealth of information that you can get by looking at very, very ancient conserved proteins, but looking at it across evolution, how they have evolved to deliver their functions in different genomes. So I will tell you the two different splicing factors, and I will not at all talk about any of the work that we did in budding yeast. When we came back, we started a lab in budding yeast while I was still figuring out what would be my big questions in plant biology. So I won't talk about any of the work we did in budding yeast. They're all published in Genetics, NAR, JCB, and the first paper in RNA from this country was all our budding yeast work. But then I said, okay, yes, all this is very exciting, but how does this impact what we know about other unicellular yeasts and fungi? And that's when Amar Clark came here for Sharath Chandra's Festschrift 60th, and he said, Usha, why are you not doing stuff in fission yeast? You should. I said, but Amar from Cold Spring Harbor, and he's the one who discovered the mating type locus and mating type switching triggered by a very specific cut and so on. So he said, no. I said, I don't know how to do the genetics. And he said, come. And he just took me to the microscope and he showed me how to do tetride dissection. And then he said, you want anything, you go to IMTech. Singh is there, you can use his thing. And that's how I jumped into it. So this is for the students in the audience. Don't be afraid to jump into fields in which you have not worked on before. There's always stuff to discover. Then, about 7 years ago, I did work with Cryptococcus. Now this again, I wasn't trained to do any work in Cryptococcus at all. But the questions led me to Cryptococcus. So I walked across to Kaustuv at JNC, and I said, you know what? These are the type of things I'm thinking of. How the hell will I learn how to do anything in this? He said, don't worry, we've got the tools. As you move along, you'll do it. So we'll tell you how we've discovered things in fission yeast and Cryptococcus for two splicing factors, which are very ancient in the control of gene expression. Now, this is all textbook stuff. So how do you express genes from eukaryotic genomes? Of course, you have this big engine, which is the transcription machinery, which is controlled by the activity of transcription factors that decide where to locate these RNA polymerases. You get transcription, you get your nascent RNA, which could be a protein coding RNA. It could be at that time when we discovered introns, we did not know that miRNAs existed. They're there in long non-coding RNAs. Any Pol II transcript can have introns in them, which need to be processed by the precise recognition of these splice junctions to give you the mature RNA. It may be a mature pre-mRNA, or it might be a mature non-coding RNA, or it could be a protein-coding RNA, which is then transported to the cytoplasm. And in the cytoplasm, it can be translated by the ribosome, and then you get your proteins, etc. So in order to deliver to the cell's current state, whatever protein factors or RNA, mRNAs, or which will be stored and then translated later, or miRNAs or non-coding RNAs, you need a dynamic coordination between several steps, which is post-transcriptional. Yes, of course, turning on and turning off genes is important, but there are so many layers of post-transcriptional gene regulation which can be investigated. And this is even more complex in eukaryotes because there is compartmentalization, which offers you the control of gene expression. One mode of compartmentalization, which is very well recognized over decades, is this nuclear versus cytoplasmic. But now we know that there are other compartmentalizations. You have membraneless compartmentalizations, which can occur due to the sequestration of RNAs and proteins into membraneless components. So add more and more based on what we discover on how gene regulation is not just on and off of genes, it's just multi-levels. So let's go back to this very, very core, conserved, ancient, evolutionarily ancient set of two transesterification reactions, which are so precise in exactly recognizing the intron-exon borders. You take yeast, budding yeast, Saccharomyces cerevisiae, or take human, or you can plug in there any vertebrate mammalian, nascent transcript, and you can recognize that there are very, very short stretches of conserved sequences which are present at around the intron-exon junctions, this orange and gray or gray and yellow, and a stretch of sequences which are present just upstream to the 3' splice site. But these are conserved across eukaryotes, but their signatures are very, very loose. And in the best case is in budding yeast, where you have an hexanucleotide, you have another hexanucleotide, and you have a trinucleotide signature, which defines the 5' splice site, 3' splice site. And the 3' splice site is also defined by this branch sequence. And in the branch, there is an adenosine, which is 100% conserved. So this is the branch side adenosine. The chemistry is very highly conserved. It's a set of two transesterification reactions where this branch adenosine 2' hydroxyl group attacks the phosphodiester bond between the last nucleotide of the exon 1 and the first nucleotide of intron 1 to give you the intermediate species, which is a 2/3 molecule, where the intron is still with the phosphodiester bond with the 3' exon, and the free 5' exon is present. This is followed by a second transesterification reaction where the 3' hydroxyl of the 5' exon attacks this splice site to then give you your spliced or ligated exons and your lariat intron. So the chemistry, this two-step transesterification chemistry, can occur in free of proteins. But the complexity of this two-step chemistry happening is complicated by the fact that the RNA doesn't have a structure to bring into proximity these splice sites such a way that the transesterifications can be precise. So the functions of taking a very, very loosely structured RNA, rather not very structured at all RNA, into something which is capable of carrying out these reactions precisely is carried out by the spliceosome, which is a mega-Dalton complex. And the discovery of the components of the spliceosome was done principally in budding yeast and HeLa cells by a combination of genetics, the kind of stuff that I did and Christine's lab did, and biochemistry, which John's lab did, Tom's lab did, Philip Schall's lab did, etc. that is making splicing competent S100 extracts and taking one model transcript, whether it is sp40 intron flanked by a few exons, or in the case of yeast, it was actin. So you take one model in vitro RNA and you make your splicing cell-free extract and try to understand what is the process, how do things come together in order to give you the splicing. So this is textbook knowledge, but it's always good to quickly revise on that. So all of this is based on in vitro splicing reactions carried out with mammalian extracts on a model substrate, or yeast extracts again with a model single substrate. Okay. So the first thing that happens is that you have the assembly of the U1 SNRP at the 5' splice site. U1 SNRP itself has its U1 snRNA and its complex of about 10 proteins. And the 3' end of the intron and the 3' exon is recognized by U2AF, not the entire U2, the U2 auxiliary factor, which comes in two flavors. It is actually a dimer of a 35 kilodalton protein and a 65 kilodalton protein. This are the first determinants of the edges of your intron, which needs to be recognized. This is followed by the action of helicase, which is PRP5, another helicase, which is sub2. These are involved in remodeling over here, such a way that now you can bring in U2 to the 3' splice site. This is followed by the assembly of a tri-SNRP complex of U4, U6, which are base paired with each other. This is the di-SNRP, and the tri-SNRP is with U5. This pre-assembled tri-SNRP complex joins in here to make something which is a pre-spliceosome. This is a transient complex which actually contains all the 5 SNRPs present in there. This SNRP, pre-spliceosome is remodeled by another ATPase, which is an RNA-dependent helicase ATPase, PRP28, to give you the B complex. And in the B complex, you have another set of huge protein complex, which is the 19 complex that comes in. Together with this, for this remodeling, you need another helicase, which is BER2 helicase, that comes in, kicks out 2 of the SNRPs. It removes your U1 and it removes U4. What is the key thing? Why is U1 and U4 removed? U1 is removed so that now the 5' splice side can be dynamically base paired with U6. And the timing of this base pairing interaction between 5' splice side and U6 is very, very important. And what is U4 doing? U4 is keeping U6 away till it can be delivered. So it's the carrier molecule. U4 is the carrier for U6, and U6 needs to be at the heart of the catalytic center which is being built, and that delivery is done by this di-SNRP. So once you have it, now you have something which is an activated complex. Still no splicing has occurred. Up till here, and all these complexes were identified either by gradient centrifugation on glycerol gradients or by native page on 3% native page gels, and these complexes range from 2 megadalton to 3.5 megadalton. So we identified all these complexes back when I was a PhD student. So of course, my role was I made mutants in the splice sites, and I discovered all of these factors, which I'm going to talk about now. So when you have this, you remodel the spliceosome again with PRP2, and that allows for the first step of splicing to occur. And then after that, the second step occurs again by another remodeling. And after the second step splicing has occurred, you have the spliced complex as well as the intron, radiate intron, which is retained in a post-splicing complex called the P complex. And the P complex is then disassembled by another RNA-dependent ATPase helicase, which releases out the spliced mRNA. The intron is then disassembled by a D branchase, and after disassembly, these splicing factors are recycled. So this entire round happens for each and every intron. That's what the in vitro splicing reactions showed. And to an extent, some really nice work that John himself did after he retired from Caltech and moved to UCSF, He actually showed in Nature Structural Molecular Biology using FRET, by putting donor and acceptor labeled nucleotides at specific positions, that in budding yeast, largely this is true. There's a stepwise assembly. But there are evidences also that the spliceosome could be pre-assembled and then loaded onto the transcript to carry out splicing, which need not be post-splicing, etc. So now let's just focus on these two factors, couple of factors which are close to my heart, discovered amongst the 17 that I found when I was a PhD student. So we'll talk on SLU7 and we'll talk about PRP16. So what are they doing? So PRP16 is a helicase, which is required for the remodeling in order to move the spliceosome RNA configuration at the catalytic center from one where the first step has occurred to one where the second step can now occur. So that's what it does in Saccharomyces cerevisiae, right? And the in vitro splicing with sp40 introns, etc., shows in mammalians also it does the same thing. We'll also talk about two other factors, that is PRP18 and SLU7. I think I'm not talking about PRP18, just SLU7. And SLU7 is a part of the U5 SNP, and that is required for 3' splice site definition, which is where the second step reaction is going to occur. The second reaction transit is going to happen at the 3' splice site. So both these factors are in Saccharomyces cerevisiae join the spliceosome after the activation of the spliceosome has been done, after all the complexes are put together and it's been activated by the BER2 helicase, etc., to then just do the second step, right? So quite specialized functions in performing the second step of the transcription. Now that's what you know from Saccharomyces cerevisiae. But to all intronic signatures, I told you that the best conserved sequences are seen in Saccharomyces, where you have this hexanucleotide at the 5' splice site, you have the trinucleotide at the 3', and then you have this branch consensus point. The only thing that is really, really conserved across species, so you're going here from Cerevisiae to humans, flies, Cryptococcus, Aspergillus, Arabidopsis, you can throw rice, whatever. It are the only two nucleotides at the 5' end of the intron, that is GT or GU, and the A, 100% invariant, and your AG. Oh, okay. No, now, so thank you. So this is the only signature. Now, when you look at, even compare in fungal system, you compare Pombe and Cryptococcus and Aspergillus, there's already a huge amount of degeneracy right at the 5' splice site. And add to that, you will see that the percentage of introns which are present in these fungal genomes is very different than that of Saccharomyces cerevisiae. In Saccharomyces cerevisiae, there are only 250 genes which contain introns. But splicing is still essential because of those 250 genes which contain intron, more than 200 of them are present in ribosomal protein coding genes. So even if one of them is not spliced, you have a problem. So splicing is absolutely essential in Saccharomyces cerevisiae. And about 20% of the transcripts which are made in Saccharomyces cerevisiae contain introns. Why? Because ribosomal proteins are transcribed in bucket loads. So it is very important. So 1 in every 4th transcript requires splicing. But if you look at other genomes, the need for splicing and the huge dependence on splicing comes from the fact that the introns are present in larger number of genes in the genome. And if you look at Pombe, it's about 43% of the genes which have it. And secondly, these introns in the Pombe as well as other fungal genomes are very, very short as compared to the human genome, where the average size of these introns in the human genome is around, I think, 3 kb or something, 3,000 nucleotides. That's the average. You can have mini introns and you can have huge introns. You can have this, but the average is around 3,000. But you look at the average in Saccharomyces cerevisiae, it's a sort of bimodal distribution. You have short introns which come around 100 or less, and you have some long introns which are 200 plus. So Fink's hypothesis is that these are busy getting eliminated, and all these introns are going to disappear from Saccharomyces cerevisiae in some time. Okay. Now you look at Pombe, Aspergillus, Neurospora, Cryptococcus, all of them are extremely short. They are less than 100. You don't have this large fraction at all. And another thing is the atypical positioning of this polypyrimidine tract. So this is another loose signature that's present between the branch point and the 3' splice side in almost all the introns. But you look at these fungi, you see that yes, they do have near the 3' splice side, they have a polypyrimidine tract, but a lot of them have a polypyrimidine tract near the 5' splice side also. So the signatures which are present are loose, the sizes of the introns are small, and some of the signatures which are normally present only in the 3' splice side are also found in the 5' splice side. So what does this mean? So genome sequence analysis of Pombe threw open the field a lot. So after Cerevisiae, Pombe was the first genome which was sequenced. Amar Clark coming here, all of this, just sort of gave me a kick in the butt and said that, don't be in your comfort zone of Saccharomyces cerevisiae genetics. You know how to do that. Go and learn. That means what? I go and, tell my students, what we should be doing, Pombe. So, so life is always a learning curve, and you just cannot step back from that. The excitement is in learning. Your knowledge from the past, of course, you apply it to the future. So we jumped into this, and we said, okay, what the hell do we know about Pombe? Now, fortunately for me, I knew Judy Potashkin did exactly the same screen that I did in Cerevisiae, because she used to keep calling me from Cold Spring Harbor and saying, how did you do the screen? How did you identify? I got this mutant, this, but so all that. So Judy had identified some mutants in Pombe, and those were PRP1. SP means Saccharomyces, Schizosaccharomyces pombe. SC means Saccharomyces cerevisiae. She identified PRP1, and she identified the PRP19 equivalent, PRP5, and PRP2 was known. All of these were the accumulated RNA in the mutant was the unspliced RNA. She didn't get anything which I got. I got the ones which were unspliced. That means they were arrested before first step catalysis. But I also got mutants which were arrested after first step splicing. They accumulated the free exon 1 and the lariat intermediate. She didn't get any of them. She was, of course, she said, I'm not getting those. I said, you get what you get, maybe you have to do more. But that time we did not know that these intronic signatures in Pombe, genome was not known. So you wouldn't know. If you catch on to one lot of model transcript and do, you will discover only that. Okay. So we didn't know it was important, but this was another thing, that whatever mutants were got in Pombe were showing unspliced precursor accumulation, therefore they act before the first step. So now what can we do? So Shataparna Banerjee, Piyush was the last PhD student who worked on Saccharomyces cerevisiae, and he helped a little bit in this fission yeast. So Shataparna, I said, okay, you know what? We should be looking at SLU7 and fission yeast and see what it's doing with respect to 3' splice site definition. Why? Because the intron sizes in Pombe are short. It's much shorter than Cerevisiae. It's got these atypical polypyrimidine tracts. It's got degenerate 5' splice site, 3' splice site. So in Cerevisiae, it joins the spliceosome late as a dimer with PRP18. SLU7 is a U5 associated protein. And this is work from David Horowitz at Maryland, who showed that this its role is to form a bridging interaction between U5 and the 3' splice site. So what did we do? There were no mutants. I just told you that Judy didn't get any mutants. She did what she thought was a saturation mutagenesis. But SLU7 in Saccharomyces is an essential gene. Right. So what we did was we constructed conditional expression systems where we have a thymine regulated promoter, which when you add thymine, it will repress the expression of this promoter so that you will not get the expression of the gene that you put downstream. So we put SLU7 here, and we integrated this at the LU1 locus. This is the normal locus that's present. So in the wild type strain, when there is no thymine, you have slight overproduction of the protein because you have both the copies present. And then when you add thymine, you shut down and you have the normal levels of expression coming from the chromosomal locus. Then we also generated another strain which was deleted for its endogenous locus, but was kept alive because we had integrated into the Lu1 locus an extra copy of the gene. And now we could bring in either wild type or mutant. So if it's wild type, you have wild type overexpression without thymine, or mutant version or overexpression without thymine, the cells are kind of alive. But if it's mutant and there is thymine and the reduction of protein level, the cells start dying. Okay. So this allowed you to play with what type of protein you want to express from the Lu1 integrated locus. So we did all of that, and what we did was we got Agilent to make splicing sensitive microarrays to our design. So we curated the entire genome's introns, made each junction-specific oligoprobes, got them printed on slides, and did the analysis. In wild type, okay, so we did it in wild type, and we also did it in a missense mutant, which was identified to be in a residue that creates its instability. And this instability is because it lies in a hydrophobic residue predicted by Raghavan Varshney. So you need to be able to traverse from genetics to genomics to biology of protein folding. There were no mutants known. And the only signature was a zinc knuckle, and the zinc knuckle mutant was dead. So dead tells you it's a very important process, but dead doesn't tell you how does it work, right? So we used Raghavan's protein folding algorithm to predict buried residues, made number of mutations and number of buried residues, and this was one buried residue mutant that was alive. So this is what we kept integrating into the Lu1 locus. And we did this splicing sensitive microarrays. Forget about the microarray, but I'll tell you what the result is. So if SLU7 is an essential splicing factor, is it essential for each and every splicing reaction? Remember, I told you that the spliceosome recognizes splice junctions intron by intron, intron by intron. In a transcript with 10 introns, it has to do this 10 times. It's not like done and dusted if you do one. Okay. And it's not a pre-existing complex, the in vitro work is telling. What we found was, yes, it's an essential factor, but there is about 15% of the entire genome's introns, splicing is not affected in this mutant. And there are two signatures. Many of them show 55%, that's a majority, again showed what Judy Potashkin saw. That is, you have no splicing at all. Whereas from Saccharomyces cerevisiae, the prediction was you should accumulate the lariat intermediate and free exon 1. You lost SLU7, right? So the phenotype of the cell is that it is essential, very good. But its impact on splicing is that it's not essential for each and every splicing reaction. And the majority of the arrest is before any splicing has occurred. That is before first step. Okay. So then having this allowed us to bin into different kinds of transcripts which are spliced independent of SLU7. What is present in them? Why are those introns being spliced even though SLU7 has been depleted? We found that it is independent. If you increase the distance between the branch point and the 3' splice side by inserting this, it makes it dependent. So this is a feature that contributes to the dependency. And another one, we found that regardless of it, it can be dependent. So it's not that only one feature of the intron correlates with dependency on the splicing factor, right? So it is a multi-parametric thing where both the distance between the 5' splice and 3' splice side, and in many cases swapping sequences from the 5' end of the intron, making all these chimeric transcripts, I'm not even going into it, several different chimeric transcripts were made to tell us there are one or two features which are important. One is the distance between the branch point and the 3' splice side, and two is the presence of this extra polypyrimidine tract which is near the 5' end. Okay, so genomes are evolving. The spliceosomal proteins are also evolving to recognize these new signatures and promote splicing. So we showed that SLU7, which is a second step splicing factor in humans and in yeast, based on in vitro splicing assays, I'm saying on one transcript. Maybe similar things are happening, again, transcript and intron specific, even in the human genome. But that hasn't been gone to that level of detail. So what we show is that the major arrest is before first step. And we had a whole bunch of genetics done to show that it interacts with U5. We have synthetic lethal interactions with U5 snRNA loop 1, not showing you any of that, and synthetic slow growth with PRP1, which is a component of the tri-SNP complex. So the genetics shows that it's interacting before any catalysis, and the majority accumulation phenotype is before this. So this was our first major paper in fission yeast, getting into after the fission yeast genome came out and using genetics and genomics to figure out that a very, very conserved protein, SLU7, evolves as the genome is evolving. So the second protein that we took is PRP16. And what is PRP16? It's a dead box RNA helicase. And its role is in proofreading of the branch side and allowing the remodeling to move from a conformation, which is first step splicing dependent, to the second step. So in the first step, what are you doing? You need the branch nucleotide to see the 5' splice site and attack there. Then you remodel it in such a way, now you trap the exon 1 and you tilt the exon 1's 3' hydroxyl to be facing towards the 3' splice site. So that remodeling is the time when PRP16 dead box helicase is reading the branch conformation, is reading the conformation of the branch nucleotide and saying, okay, yes, everything is all right, you can proceed and do the second step. If things are not okay, you can backtrack. You can disassemble. And if you disassemble, you will kill, because there is a lot of NMD options available, and decay pathways are available to get rid of the unspliced RNA. Right? So PRP16 is a very important proofreading enzyme. And PRP16 came out of my genetic screen, but it also came out from Roy Parker. Roy Parker's postdoc is the mentor for Raj. Roy and I published my first splicing papers with Roy. Roy didn't discover PRP16, I should correct myself. It was Sean Burgess who set up a screen to look at suppressors of branch point mutations. And he landed up with PRP16, which he called, I forget what, some Burr, maybe Burr 1 or something. Burr for feeling cold in the cold room. Okay, brr. Anyway, so and I got PRP16 for pRNA processing mutant number 16, and all the factors post 16 came out of my screen. So we discovered 2 different alleles, etc. So Beata Schwer and all these people showed what PRP16 does and the proofreading functions. So what is PRP16 in Bombay? What is it doing? Okay, so like a dead box helicase, it's a respectable protein. It has all the ATP binding and hydrolysis motifs. It has the RNA binding motif. It's got all the necessary signatures, right? And properly positioned, everything is fine. So we said, what does Beata Schwer did a lot of mutagenesis on PRP16 and Cerevisiae. Let's look at those mutants and pick a couple of positions where we can make mutations which are not going to be lethal, but which were conditional in Cerevisiae, so that we can get conditional mutation. Okay, 16 knockout in Bombay is dead. Okay, so knockouts are dead, so we have to do something else. All right. So we chose this, do you see this phenylalanine tyrosine? This was shown to be important in PRP16 of Cerevisiae. And we also chose this first residue over here. Okay, now we made these mutants. And again, what do? We knock out the chromosomal locus, endogenous, and insert into the Lu1 locus whatever we want to do. We can insert a wild type extra copy, or we can enter the mutant extra copy. And then we can use this thymine switching drama to see what is happening, right? So what do we see? PRP16-1 allele, which is the F528 to A, if I'm not mistaken, or H. We had many substitutions there. When you are growing it in complete medium, that means it's healthy and happy, lots of media, nothing happens. But the minute you start moving it into non-permissive conditions, that is, you have thymine and you increase the temperature, this one is dying, but the other allele is kind of growing. It's not that unhappy. It's all right. So here we got two alleles, two different regions of the protein. One of them is okay and one of them is not. So again, I'm skipping a whole lot of stuff. Again, we made POM-based specific splicing arrays. We started making it. This is when NGS came. Okay, forget that stuff. Learn to do NGS. Learn the bioinformatics. Find people who can teach you. You just have to do it. So we jumped into NGS. Okay. So, and what do we have to do? We wanted to find out what is the splicing index. That is how for each intron we compute how much of exon intron 1 signal is coming and how much of intron exon 2 signal is coming, divided by how much of the exon junction is coming. So unspliced, unspliced, 2 signatures for unspliced, 1 signature for spliced, and then compute the ratio of the 2 and derive what is a splicing index, and now plot this as a differential between the wild type and the mutant and put some cutoffs. Okay, so we did all that, and again we find PRP16 is very important. So in SLU7, we found 15% of the transcripts can be spliced independent of SLU7. Here it was much smaller. It was about, I think, 8 to 10%, smaller than that. But still, there were small number of introns in the Pombe genome. Pombe genome, 43% have introns in them, were spliced independent of PRP16. So we started, so I mean, that's what this is. Okay, F528S, spliced beautifully because no precursor in the wild type, no precursor in the mutant. You look at DGA1 or SEB1, you start seeing these unspliced precursor. So these are things that we pick up from, I'll stop at the end of this, okay? All right, so that this is just proof to tell you that when I'm saying it's spliced and unspliced and all that, it really happens. I mean, we've got dozens of these done. Then we said, okay, what are the signature features? Is there something which is in these versus this, which tells you that things are different? So the strength of interaction between the 5' splice site and the U6 snRNA. Now, this is a branch site recognition residue in Saccharomyces cerevisiae. But what is it doing? It's looking at this interaction. So somebody in one position doesn't mean that they're fixated on that. So you, when you're such a short intron, you will look here also, you look there also, you look here, you look there. That's what's happening. Okay, so PRP16 is gauging the strength of the interaction in the 5' splice site, +4, +5, +6. You can see there is degeneracy here. But all of these have to make some interaction, base pairing interaction with the Akaga box in the U6, and the Akaga box, this ACA, 100% conserved in all species. You take plants, it's there. You take humans, it's there. That is in the catalytic center. So Akaga box of the U6 is invariant. But this 5' splice site is sloppy. So this sloppy pairing, the stronger it is, the better it is. The weaker it is, then PRP16 has to come, and its functions are important for this stabilization. How do we know that? We made mutations in, again, we took a transcript and made, first we did all the bioinformatics to say what happens if all the 3 are strongly base paired, then you can detect the dependence versus independence. So all these transcripts are made and you have mini genes and you test them out. Okay. So the strength of the 5' splice site U6 interaction is important. But that didn't explain all our model transcripts, which we were testing. So we said, what's going on at the branch site and the U2? Because branch site is supposed to be where PRP16 looks. That's what Sean Burgess and Beata Schwer and all that said. So we looked at the adenosine over here. Yeah, it's perfect. It's 100% conserved. But around the adenosine, what is the snRNA that's present? It's the U2. And I think this is U2 helix 2AB or something like that. So I forget the details. And there are a number of base pairing interactions. Some of these are not perfect, but they do occur. Now, this is also a determinant. So if you look at the -4 position of the branch site to this, you start being able to discern that this is also a signature. So you have not, PRP16 hasn't changed its character totally. It is still scrutinizing the branch site. It is scrutinizing the branch site, and signatures over here, as well as signatures at the 5' end, both of them are required for its function. So we not only did that, we made mutants in the factor. And in Saccharomyces cerevisiae, if you make a branch site A to C, this is the Burgess suppressor mutant which was done. If instead of A you have a C, then CWC25, which is a first step splicing factor, doesn't get kicked out and is sitting over here. And even though you have PRP16, nothing much is happening, and the unspliced pre-mRNA is moved into degradation. This is in Saccharomyces cerevisiae. But when you have the C2A and you have a mutation in PRP16, this becomes a sloppy helicase, doesn't do the proofreading, and because it doesn't do the proofreading, it allows to go through splicing, and you use a C residue which should not be used. Okay, so the proofreading capability of PRP16 was proven by making this type of mutant. So we made those type of mutants, this, that, I won't go into the description, and we showed that in Saccharomyces cerevisiae, the CWC25, that's this character, that is scrutinizing, sitting on top of this A residue, and then PRP16 comes in, reads that, oh yeah, everything is okay, go forward. ATP converted to ADP, helicase, restructure model, kick out CWC25, allow these second set splicing factors, including SLU7, to come in. So we looked at how is it positioned in the case of cerevisiae versus how is it positioned in the case of Pombe. This helix, which is at the N-terminal end of CDC25, fairly conserved. Long helix, you can superimpose it on the cryo-EM structure, which was available by then. And we looked at what is the what is the interaction between this serine versus glycine. So where is the serine? Do you see this over here? This is glycine versus serine. So if you have serine, then the distance from your branch nucleotide, which is the A70 at the branch site, is shorter than if you have glycine, it becomes longer. So recognition of this adenosine, which is right near the branch nucleotide, by this helix of CWC25, by changing just one amino acid, you increase the distance. So what do? You make that role of PRP16 even more important in scrutinizing what the residues are present at the branch site. So I will stop over here and summarize that there are early interactions in both the splicing factors. In the most recent splicing factor, which is the dead box helicase, it's both the determinants of interactions with the U5, U6 and U2, and their substrate-specific functions. So I'm not going to talk about this. So in the next part of the talk, I'll tell you what we've done in Cryptococcus.
Okay, we have time for questions.
Oh, I was wondering, did you look at alternative splicing and if that is affected by either changing the concentration of your PRP16?
Yeah, we've done it. I'm not showing you the slides. Yeah, so for PRP16, I don't remember the top of my head, but for SLU7, I remember at the top of my head. We showed that alternative splicing, even though it's very rare in the case of budding yeast, it does occur. There are 1 or 2 transcripts, one of them being the MAT-A locus. The other one is a ATS1, where there are 3 or 4 splice isoforms. We showed that weak splice sites are dependent on the splicing factor. So supposing you have 2 5' splice sites, one is a strong one and one is a weak one. What do you mean by weak? Where it is slightly off consensus, right? So in wild type cells, you will have 80% using the strong and 20% using the weak. In the SLU7 mutant, what you will find is that the weak one doesn't get utilized at all. So the utilization of degenerate or weak splice sites in a transcript which has multiple offers you the change. So this we showed is true for the 5' splice site weak one and also 3' splice site. So sometimes you'll have CAG, sometimes you'll have TAG, and you'll have two of them. So it helps in the choice between alternative splice sites. Right?
Can I ask another question?
And we also showed that that was stress dependent. What did we show? We showed something about heat stress, cold stress, cadmium, caffeine, all of that affects it. We also showed that it depends on the speed of transcription elongation. I've been in this business for a long time, so I really couldn't cover all of that. So if you slow down transcription, okay, then it starts using all these weak sites. So none of these machineries are working in isolation. Spliceosomes U1 is riding on the the heavily phosphorylated tail that RNA polymerase has, right? So it has the serine phosphorylations, etc. That's what loads on your U1 and brings it there. So if you slow down transcription, I forget that drug that we used. We did something. Titash did that. We slowed it down. Then also you will see that the percentage utilization of the strong versus weak starts changing. Yes, yes. So speed of transcription, elongation, and the presence of splicing factors changes the percentage utilization of these strong versus weak splice sites. Yeah, it will. If you can, we've done this not only for that, we've also done this for falciparum. What you need is that the helicase part of the protein is more or less similar. But many other eukaryotic proteins have a long N-terminal extension. So you need to make chimeras. If you just take it as such, it doesn't reach the spliceosome. So in order for it to reach the spliceosome, it needs that species-specific N-terminal. So we did in falciparum, we showed that if you make this helicase domain of falciparum hooked up with the N-terminal of Pombe, it will work. But if you don't do the other way, it won't. We did this work with Pawan Malhotra at ICGB. So he contacted us saying, you want to do something with this? Will you do some work with us? So I did it, yeah. Yeah. Of consensus.
Okay.
Yeah. So when you look at this signature, so this means G and T fully conserved. Now, if you come to 1, 2, 3, 4, the fourth residue can either be an A, or a T, or a C. So a C, so this is a position weight matrix for that 6 nucleotide across the whole genome. Yeah.
Generally, don't select or use under normal conditions.
It will be used, supposing this is the only one, it will be used provided the splicing factor is present. Now, if the splicing factor is not present, then it will not be used at all. And in the case of alternative splicing, which we add, supposing we had 2 5' splice sites, one with an A and another one with a C, which are right next to each other, it can happen. Which one do you choose? You use the A is used more efficiently, C is used less efficiently, but let us say under caffeine stress, you want to change the ratio of those two, then your splicing factor comes in. Yes, Raj.
Yeah, thanks for that wonderful overview of splicing, Usha, and taking us through the journey. Again, related to his question, the evolutionary significance of weak splice sites. I mean, just thinking about it, it would be nice if all splice sites were created equal.
Then you will not get diversity. You will not be able to, if every intron is spliced efficiently, then you will not be able to modulate splicing level transcript abundance. Right.
Right, but
so
these
let
factors.
Let me complete. Remember this elongation story I told you. The polymerase is a huge engine. It's just going gangbusters, right? So there will be some sloppiness in the way spliceosome will choose the sites. So you have that. And secondly, supposing you want to get rid of something, you need to be able to splice in such a way that you leave the intron in, and that happens. Not every intron is 100% spliced. You'll have a small percentage all the time which will be unspliced. So the efficiency of splicing, even in Saccharomyces cerevisiae, if you look at MAT1 locus, you will see 50% is unspliced. But that unspliced is usually taken care of within the nuclear as well as the cytoplasmic decay pathways. They're taken care of. So if you want to change a large transcript, output from a gene to a low transcript output, you have to stop that engine. That takes time. So before you stop transcription, you can dispose of these things by deliberately choosing sites which are not favorable. That is, you leave a bit of the intron in. If you use a downstream site, you leave a bit of the intron in, you throw the reading frame off, it is recognized by either UPF or other NMD components, and you dispose. So it affords you the way to titrate and fine-tune post-transcriptionally at the level of splicing. Right? Yes. And the next story in Cryptococcus, it becomes even more apparent why you have it. No, they're always recruited. They are present. Whether their action is required or not is different. No. They are functioning when things go wrong. They have to function all the time. This is essential even for the consensus element. It doesn't mean that if it is A, that PRP16 is not involved. Its role becomes uncovered when you have a weak site.
Is it possible that some of these are like
Positive.
too much concentration?
Definitely. So because if you want to keep, if everything is okay, you want the kinetics of the reaction to be go. Okay. If something is not okay in between, like your branch nucleotide is not okay, your 5' splicite is not okay, you want to either backtrack so that you can read one more time. That's your proofreading. Right. So what can you do? You can backtrack and see. That is, you go back to a confirmation. But supposing you have already disposed of some of the splicing factors. Remember, I told you there are restructuring. U1 has gone, U4 has gone. You can't backtrack all the way, right? So if really all those have gone, you can't backtrack. So the hypothesis now is that the cerevisiae model, that everything is disassembled and reassembled again, may not be true for each and every. They might be all present over there. It's just that they will join or they will leave. They'll all be in the vicinity, especially when you have a short intron. Okay.
Your genome-wide screen that you have done, where you found out these things.
Oh, that was when I was my PhD student.
I know.
Okay.
Okay, your PhD student has done. You didn't.
No, I did.
Yeah.
As a PhD student.
Try doing it under different nutritional condition, because that can then perhaps correlate some of the transcripts which are needed.
What people did, Christine's one student, Michelle something. So they took all these factors. So the thing is, I think we have saturated because people are not discovering more and more core spliceosomal factors. Okay. So she took the battery of all the mutants. We had about 20, 30 mutants. First step, second step, recycling, all of them, and tested them under different nutritional conditions and did genomics. So then again, you pick up that under nutritional stress, I said caffeine, which is an external, let's say, DNA damage triggering type of stress. So under those conditions, you may want to change the ratios or the, so there it comes in. Core splicing factors come in. So in some other cases, like in people will say that, HNRNP subtype this came and utilize this site. Why? Because it masks a good site and exposes only the bad site or a weak site. So that's how things work. Yeah, so people have done that. That is, we haven't done that. So they took the battery of mutants and then tested it under various conditions. Yeah. Sure. What if? Yeah, G also doesn't splice. They're all done 35 years ago. So yes. Yeah. So this bulging is essential, and that is a remnant of even more ancient one, which is completely group 2 splicing that is seen in parasites and mitochondria and chloroplasts, etc., which is completely protein free. Only 1 or 2 proteins are needed. In Neurospora, they showed that 2 proteins. So that helix is very, very ancient. This helix with the bulge is seen in protein free RNA catalyzed splicing. They're there. Actually, it turns out that this come out a little bit crunched. Can you see there is A, and next to it, the next residue is also an A. Okay, so all those mutants are there in my paper with Roy Parker. So we've made mutants in that, right? So what we find is that C doesn't work. Robin Reed did in Tom Maniatis' lab in the SV40. Myself and Roy, we did it in the Saccharomyces cerevisiae using the actin intron, right? So A to C, I showed, that was my first paper, if I'm not mistaken. Little bit it will utilize. A to G, it will not touch at all. So we did map the branch site, did T1 RNAs, nucleotide fingerprint, and sequenced the branch, found out the branch, and actually did analysis. So C will be utilized a little bit. G will not be used at all. A is highly preferred. So we did all that. Many factors are there. So in Bombay, many times the homologies of the splicing factor to the human is much closer than cerevisiae. And you often have more number of these auxiliary factors, more copy number, etc.
Okay, let's close the session. Second part of the talk.
Okay, I'll take a drink of water.
Yes.
Okay, so, all right, we were having a lot of fun doing this stuff in Bombay. Everything was all very good, this and that. But, then came genome sequencing became so easy that many genomes are being sequenced. So not only Bombay is related clade of Japonicus, etc., all were being, roughly in the Schizosaccharomyces genera, it's about 40 to 50% of the genes had intron. Then came the sequence of Cryptococcus neoformans. I should have italicized, sorry. Cryptococcus just blew the whole field open. 99% of its genes contain introns. It's perhaps even a little more than even the human. Okay. And another one is many transcripts have many introns. The average number of introns per transcript is 5.5, average number. Okay. All the introns are tiny. The average size of the intron is 56. So splicing is a huge task in Cryptococcus neoformans. 99% of the genes have introns. Most of the transcripts have multiple introns per transcript. Some of them have even 20 introns. So look, tiny introns which are present. So this is an extreme case of splicing by intron definition. So in mammalian, Sridhar will know, the problem is in identifying the exons, which tend to be small and the introns tend to be huge. So that is exon definition model. So you need to identify the end of one exon and both the ends of the exon. So that's exon definition. Whereas here, it is intron definition. So this is an extreme case when one can study what is this intron definition model and how does it work and what can we do over here. So another role why these introns are present, one is that it can fine-tune gene expression. We've been talking about it. But the other thing is that many of these introns often contain cis elements which actually help in the earlier process of transcription. So they are controlling gene expression not only at the level of splicing, but also at the earlier step of gene expression, start or stop. Then alternative splicing. It has SR proteins, and any stress, huge amount of alternative splicing is going on. Okay. How is all this happening? What is there is obviously a strong selective pressure to maintain this intron richness. We don't know, but we said, okay, this is a great model, and let's see what the hell is going on in Cryptococcus. We didn't know anything how to do it. We had to learn. Now look at this. This also another table is important. These are total number of genes, and this is the total number of introns. 40,000 introns in Cryptococcus. Why 40,000? Because each transcript has multiple introns. I told you the average is 5.5 per transcript. Second, if you look at the orthologs of the human proteins, you find in Cerevisiae, you will find this many, but in Cryptococcus, you see a lot more. So these papers told us that splicing must be important. Splicing factors are present, the orthologs of these two, that is SLU7 and PRP16, and again tell you what has happened in Cryptococcus, and these are recent papers from last year and this year. So first on, this is SLU7. SLU7 is a zinc knuckle protein, and that zinc knuckle is present in all of them. You tamper the zinc knuckle, it's a dead protein. I told you that in Bombay, we made the zinc knuckle mutant, it was dead. So that's why we had to go to buried site mutation, and we made the buried site mutation somewhere over here. I think it is somewhere over here in Bombay. Okay. So the signature of this Cryptococcus one is also similar, but the Cryptococcus SLU7 is even amongst fungi seems to be very, very distal. It's very distant from. These are your ascomycota, and these are your basidiomycota, and many of you would now be famous with mucor. How many of you have heard of mucor after COVID-19 pandemic? Many people got mucor. Okay. So anybody's heard of Ustilagomatus? Rust, wheat rust. So these are pathogenic basidiomycetes. Cryptococcus is also pathogenic, but there are strains available which are avirulent, non-pathogenic, and that's what we worked with. But these are opportunistic. These are actually quite aggressive opportunistic. So this branch looks to be quite different when you look at the phylogeny. Okay. So we learned everything from Kaustubh. We got the wild type strain from him. And what was possible in the case of Vijayraghavan was also possible here, slightly different switch. What we did was at the chromosomal locus of SLU7 of, now this is all Cryptococcus, we integrated at its upstream a GAL7 transcription regulation circuit so that under galactose, this cassette will be, the normal gene will be expressed. But we also made it tagged with mCherry so that we will know whether the protein is being made or not made, etc. And use this, integrated this into the SLU7 locus, so the rest of SLU7 is perfectly normal. So now we have a strain where under galactose, it's functional. You look at the normal growth phenotype, then we put it under glucose, there'll be no expression, and we start asking what is the phenotype. So we have a conditional knockdown strain, and the mutants were all made by us. We got the wild type from him and the backbone construct to make the knockout. So what we found is when it's conditional knockout, the growth is very poor in media as well as in broth. So that's your wild type grown in galactose or glucose. The two, it actually likes glucose better than galactose. So that's your yellow and your green. But you look at your mutant, that is your purple, it's down here. And when it's on galactose, it's down here. So it's extremely slow growing. And the zinc finger node mutant makes it dead. So that was no use. Okay, so we had to work with what we had. So what we did was we started asking, why is it slow growing? We did a whole bunch of cell cycle related work in Bombay. I didn't talk about it, given that this is an RNA meeting. So here we saw what is the cell cycle effect, why is it growing slow, and we found that it was arrested, or very, very greater proportion of the cells are in the G2M after you transfer them to the repressive media. Sorry for the spelling error. So now we dug a little bit deeper, and what we did was we constructed the conditional knockdown of Cryptococcus SLU7 in strains which were differently tagged for different readouts for mitosis. So histone H4 will tell us about the nucleus. CENPA will tell us about the CENPA binds to the centromere kinetochore, so it will tell us about the kinetochore. We did it with TUB1 that tells you the spindle, and we also did it with dynein, which I'm not showing. That all of this was done. I'm not showing you anything. I'll just tell you that it leads to, so this was used for us to track the nucleus and track the spindle and track the kinetochores. That is this. Why did we want to do all of this? Is because Cryptococcus has a very weird mitosis. So not only is its genome weird, its mitosis is also weird. So Kaustubh's lab, as well as Joe Heitman at Duke, showed that it's a very funny mitosis in that in Saccharomyces cerevisiae, once the nucleus has been duplicated and you have your spindle assembling, then you have mitosis which occurs at the mother-daughter bud neck. But in Cryptococcus, the nucleus moves entirely into the daughter. The undivided nucleus with its duplicated DNA content moves from one house to the other house. It moves into the daughter. Typically, you will do the division right here and then you pull into the other in the case of all the ascomycota. Here, it goes completely into the daughter. Division occurs in the daughter and then the segregated nucleus comes back to the mother. Very abnormal, very weird, but Crypto does this. Okay, so Crypto does this, and I think Ustilago also does this. So there's some funny feature of Basidiomycota. Maybe Mucor also does it. We don't know, right? So and there are different ways of monitoring it. So that is why we use the CENPA to monitor this. We used the spindle to monitor the spindle. We used TUB and so on. So what we did was to look at what's going on with mitosis, and the sum of the substance was, does it affect nuclear migration to the daughter? Is kinetochore clustering affected? Is mitotic spindle elongation affected? Where is mitotic spindle form? So in Crypto, the funny thing is that the kinetochores are dispersed all the way until it comes to the close to the G2. Whereas in Cerevisiae, it will be clustered right here. So weird things. They are weird because we haven't looked at them. So everybody assumes that everything is just like what it happens in Saccharomyces cerevisiae. It's not. So what we did was we started looking at when does the nucleus move to the daughter in the wild type, and when does the daughter move in the mutant. Which mutant? Our SLU7 knockdown mutant. Right? So here is live imaging, and you can see that the mother, it's mother cell with the marked nucleus, which is this H4 marked, if I'm not mistaken. It goes around here. It takes about that much time. And within this, if we mark this to this, all the division has been coming. Okay, see over here, got completely into the daughter, then pulled back into the mother in this division. So that's your kinetics. 3 to 4 minutes, 5 minutes, it's done. The mutant, you find most of the cells, it's sitting like a rock. Nucleus has finished G2. We can tell that by facts. The DNA content is diploid, but the nucleus is not moving. Okay, no nuclear movement. So then we said, okay, let's now start looking whether that is really the complete phenotype. So because this was a knockdown, so you will have some amount of the protein which is present, because we're going from an on state to an off state, right? And the on state is actually under galactose, so it's a little bit overexpressed. So you have a lot of protein, and these proteins are quite perdurant. Their turnover is slow. So they're very stable proteins. So therefore, we could actually see what are the different types of phenotypes which you will get, because protein might have turned over in some, may not have turned over in some. So we were able to see that there are many phenotypes. So in the wild type, in 4 minutes, the job is done. The one case that we found is it takes at least up to 7 minutes. So here's the nuclear migration has occurred, but it's taking longer. Then we have other cases where nuclear migration has not occurred at all, but division is occurring completely in the mother, abnormal. And this is the predominant case when nuclear migration itself doesn't occur at all. Why these nuclear migration defects in a splicing factor loss, right? So there can be two different reasons. So all of this was quantified and we were able to show that the bulk should be like this, that's gone down, and we start seeing all these three other abdominal classes. Okay. So nuclear migration defect could be the reason that there is a cell cycle defect. Okay. So now this nuclear migration defect, I'm not relying only on the nucleus, we also look at the spindle. How do we look at the spindle? Because we have done this knockout in the TUB1, which is encoding the alpha tubulin. We can now monitor the spindle. So this, you can see over here, before the spindle is properly polymerized, it's dispersed, spindle is about to be formed, spindle is formed, it's short, the entire spindle goes into here, it is in the daughter, spindle elongates, anaphase is happening, it's pulled back into the mother. Spindle elongation, it's a partially open mitosis. Spindle elongation is correlated with anaphase. Okay, so the spindle, short, tight spindle is metaphase. These are all still in metaphase. Now over here, elongates, pulls it back, and this is completed in the wild type. And in the mutant, you see all of this, you see it comes here, and it's starting to elongate right at the neck, or sometimes it's elongated even in the mother. So not only the nucleus is not getting where it should be, the spindle elongation is also wrong. The temporal control of when things should happen for a cellular process like mitosis is affected in the splicing factor mutant. All of that was also quantified. So then we did dynein. Now, why the hell should we do dynein? Because dynein is a key motor protein that is attached to the cortical sides by NUM1. This is what Umesh, Sachin's protein, NUM1. So this, it gets into the core of cell biology. What is this splicing factor doing affecting the core of cell biology? So what does it do? It goes and attaches in the daughter. So it is sitting, riding over here, goes there, makes sure that this astral microtubule touches the cortex. And when it touches, it pulls the nucleus and division occurs, right? In Cryptococcus, it's all scattered, goes in here. Look at the length of these microtubules. These are astral microtubules which go out, and riding on them is dynein, and it goes in there, and then there is this splicing. Then there is this division. So we look at dynein in the wild type, you see dynein in the large bud, you see this here, and in the mutant, you see that the dynein is now completely in the mother, or it is not there at all. So we used to discuss a lot with Sachin, and he was so excited by seeing this. He said, look, we never thought that we are going to run into looking at the cell biology of nuclear segregation. And we got some very exciting results from that. So now the question is, why is this happening? So there are two ways. One is your splicing factor could be important for the splicing of cytoskeletal elements, genes which are cytoskeletal elements which are containing introns in them, because I told you 99% of the genes have introns in them. So do they have introns in them or other cell cycle regulators which have introns in them that are particularly dependent upon our splicing factor? And when you lose a splicing factor, you're going to have all these problems. So we did deep transcriptomics. So this is, so you go about to discover all the splicing events, including alternative splicing. We did extremely deep sequencing, and we found huge number of cell cycle factors are deregulated. Some are very low and some are very abnormally very high. Very high, for example, MAD2, which is a checkpoint. So we took two of them. One is CLIN1, which is a cyclin, and PAC1, which interacts with dynein. And these are other examples of where they are differentially expressed. So now we'll tell you why PAC1. So PAC1 is this green little protein that sits together with dynein And it's pulling these astral microtubules to interact with num1, which is present in the cortex of the bud. And num1's it's Numa and all of that in other species. So what does it do? It pulls it, attaches it to the cortex, offloads after this anchor has occurred, and only then the nucleus can be pulled into the daughter. I'm showing you.
Okay, but now you right now it's different.
I'm showing you the PAC1 is one of the culprits, but it's not the only culprit. Okay, so that's PAC1. Okay, so PAC1 has, so the hypothesis is that because PRP16 is an essential splicing factor and the genome is riddled with introns, there must be some cell cycle related factors. So many cell cycle related factors are there, but we zeroed in on this mitotic defect that we saw, that is nuclear migration. So what are the players in nuclear migration? So we looked deeper into that, and we found that this PAC1, which is necessary for dynein to attach itself to the astral microtubules and pull the nucleus, that has a whole bunch of introns in it. And many of those introns are PRP16 dependent, right? So, so shown over here is one of those introns. You can see this PAC1 intron 1, unspliced mRNA, spliced mRNA. In the wild type, it's very, very less. So this is what I meant to say, that even in the wild type, not 100% splicing, there'll always be some amount of transcripts which are in the process of splicing, or it is slightly slow in the process of inefficiency of splicing. But in the mutant, you see this. So you quantify the mRNA, the spliced mRNA, and you quantify the unspliced mRNA, and you see this. So this is also there for intron 2, so at least 2 introns. But intron 3 is much weaker, so and intron 4 is even weaker. Then 5 and 6 also was done. To cut the long story short, it has 6 introns, if I'm not mistaken. Yeah, there we go. 1, 2, 3, and 5 are dependent, but others are not. So even within a transcript, you can have multiple introns. Some are dependent on your splicing factor, and some can be spliced independent. Again, why? Because there are signatures. Each intron carries its own signature elements, right? So even within an intron, you can define all of this. Then we said, okay, if PAC1 is so much important, if that is contributing to this phenotype, that is this nuclear migration, let's try, I'm not showing you the data, we put in a strain where all the introns of PAC1 were removed. Okay, and we drove it out of an H3 promoter. So we integrated and generated a strain. Now there's plenty of PAC1, right? No splicing. Did you recover the phenotype? And we showed that the nuclear migration phenotype is rescued, but other cell cycle phenotypes are still there. Cytokinesis defect is still there. So PAC1 is responsible for, so this is how, by fine-tuning the expression level of even 1 or 2 transcripts, you can contribute to a biological process, right? And imagine your core splicing factor is present in the spliceosome. It's not doing this just for nuclear migration, it's doing it for everything else also. So that is how the impact of efficiency of splicing on cellular process occurs. So we showed that if you are efficient for the splicing of PAC1 and you have SLU7 present in there, then this PAC1 is made, it loads on over here, pulls these astral microtubules, attaches them properly, brings the nucleus completely into the daughter, then spindle elongation occurs, and by spindle elongation, anaphase is completed and everything happens, right? But if you compromise over here by inefficient splicing, meaning PAC1 has at least 3 introns that are slightly weak. And if you do not have SLU7, their recognition becomes inefficient. So the amount of PAC1 protein that you're going to make is less. And when you have less PAC1, entire process is weak. The nucleus never migrates to the thing. It's in the bud neck and does this abnormal mitosis for Cryptococcus. It's abnormal. Entire division sometimes happen within the mother cell, and the daughter is an empty house with no nucleus. Okay, all of these are hallmarks of what can create antifungal resistance. Aneuploidy, antifungal resistance, all of this can come just simply because you didn't segregate your chromosomes properly, right? So that was SLU7. Now, of course, we did all the alignment for sequences, looked not only at PAC1, look at CLIN1, asked what are the features, and first story was not at all encouraging. Nothing is different between the global consensus of the dependent SLU7 versus independent SLU7. So this has all come from very deep transcriptomics. So we are assessing each and every splice site's consensus element. No difference. 5' splice site is the same, 3' splice site is the same, branch point is the same, polypyrimidine tracks are similar, maybe a little bit different in your independent category. It seems to be a little bit weaker. Do you see the number of Cs over here? So the weaker polypyrimidine track seems to be KIT SLU7 independent, and nothing which is striking like we saw in the case of Bombay. So then we said, okay, let's look at distances. Okay, these elements are not standing out. You have this abnormal polypyrimidine track here, and if that track is a weak track, then it tends to be SLU7 independent. So we said, let's look at the distance. Distance between the 5' splice site to the branch, the total intron length, distance between branch to 3', what is the AU content of the intron, and did all of that analysis. And what we found was if in the case of Cryptococcus, this distance between the branch and this is greater than 16, that means it has to be a long intron. It's dependent. Most introns are short, I told you. So if you're slightly off the average for your genome, you will be inefficient, and your splicing factor better be on top of the game. If your splicing factor is not on top of the game and you have a slightly off feature, you are going to be in trouble. Your splicing will be inefficient, right? So now here are 3 different fungi. Sorry. Almost 400 million years of evolution, 600 million years of evolution. Pre-mRNA splicing is required. Factors are all present, but the recognition of the cis element and the role of their splicing factor is what is evolving. So the common feature seems to be for SLU7 that this 3' end is very important. In Cerevisiae also, if you increase the distance, it becomes dependent. In Bombay, we showed if we increase the distance, it becomes dependent. In Cryptococcus, we didn't manipulate, make mini transcripts, but we know from the genome that this is important. So this seems to be a very, very deeply conserved scrutinizing element that SLU7 does. It looks at this distance between here and here. It's not like a strict ruler, but it is looking. Okay. But in other genomes, it's also looking at the 5' end. In Bombay, we know that it's looking at the AU content. In crypto, we know it's looking at the distance again. So the minute this is greater than 60 and this is greater than 16, this is an abnormally long intron for crypto, and it becomes dependent. Okay. So I'll finish off with the helicase. I'll only give you summary. So you got the gist of it. We pick our favorite factors, million to almost 1 billion years old, and we look at different genomes and see what do they do in that genome. Is it copycat for them or they're changing? They're changing because the genomes are changing, and they're changing in very, very subtle ways. Okay. So now PRP16, which is that helicase, right? So this was, again, we constructed the strains where we integrated into the native locus, GAL-driven, mCherry tagged, and we showed again over here that if you shut down the expression, growth is very, very slow, right? So how do we do? We grow the primary culture in permissive media, then we move it to non-permissive media, and then serially dilute, and then we look at the transcriptome, and I'm here showing you only one to show that, yes, this is your wild type, no unspliced, the knockdown unspliced, complemented, put it into the safe haven locus, it's complemented, so all of this is dependent. In SLU7 also, we've done the complementation. Right? So then we did the genome-wide splicing, very, very deep sequencing. Okay, so this is typically people will do 2 pass, 3 pass, 4 pass, and they say, I did sequencing. Very good. We do at least 10 to 20 times genome depth, sometimes even 60 times the genome, because we want to be sure that we didn't miss anything, right? So very, very deep sequencing. So you give it to a company and you say it is a fungus. And I say, I want 60 MB readout. So 60 MB readout for a fungus? There's nothing doing. You're not getting it until you give me this data. You don't give, you have to repeat it. So they are, if you want to really understand, you have to cater to that. Otherwise, they will give you some very shallow sequencing. You will never even discover things. So even in POMBE and crypto, we've done 60x genome depth sequencing, 60 MB. Really, then you can really make some big conclusions about what's going on. So we do and score what is called as IRS, that is intron retention score. So we are mapping this junction, we're mapping this junction, and we are looking at the total reads and computing what is the retention score, and plot it as a differential between the wild type to the mutant. Then we apply a hypergeometric test and do a Z-score ranking. The null hypothesis is there is no difference between the wild type and the mutant. No data. I hope you are all excited to go and read these papers of last year and this year. The summary is, what is crypto doing with its PRP16? Single copy gene, essential RNA helicase. So we had mutants in the helicase, we have the knockdown in the wild type, we have a knockdown with the mutant, and we were able to show that it is essential for the splicing of introns with suboptimal intronic features. What are the suboptimal features? Small intron, weak interactions with snRNA. And in the case of PRP16, in PRP16 of POMBE, it was looking at the branch consensus and the 5' splice site with U6. Here, it is U1 loop. So remember that the catalytic core is full of RNAs. There's U1, there is U6, there is U2. These three are the ones which are massaging the junction. So in Cryptococcus, it's the U1 highly conserved loop 1. This tetra U, very famous. Okay. And this is making base pairing interactions with exon 1 and exon 3, 5' exon and 3' exon. So this loop is keeping these two potential sites together. And it is the strength of this interaction which is determining whether it will be dependent or independent. Okay, so if you have a strong interaction, then you go through. That is, you have this, you have this, you have, so A, G, and G. So these are the last 3 nucleotides of the 5' exon. This is the first nucleotide of the 3' exon. Then you're all right even in the knockdown and you can splice. But if you're off consensus here, that is exonic nucleotides are changed, then this becomes weaker, then you're dependent upon PRP16 and your splicing is poor, and if you're not spliced, you go out. So why the hell, I mean, you're supposed to splice the intron. Why are you looking at the exon last 3 nucleotides? So this tells you that this allows for some degree of evolution to play around. Exonic nucleotides will contribute towards your protein. So you can allow for evolution of your target without binding it down. Okay, so this weak interaction tells you that it can fine-tune to accommodate diversity in cis sequences at these junctions. An exonic sequence, if you're too tightly fixed on it, then you will have too much selection pressure for those amino acids that is going to code for. So you're a little bit sloppy, but at the same time, you want to look for it. So that's what we showed in this recent paper that we published. Of course, I did very little of this work other than, I think the last time I did was when I picked up the POMBE and did the tetrad dissections and showed Shataparna and Titash to do the dissections, how to do it, how to make the strains. Crypto, I didn't touch it at all. The students did everything, other than talking to Kaustuv and telling, this is what we should be doing. They did everything. So amazing. I've listed here only those PhD students who worked in RNA and in only these two. I've not shown the ones who worked in Cerevisiae. They did an amazing job. Raghavan was really good in helping us to identify where to make the mutations when we had proteins which were dead. Shubha was very deeply involved in the Bombay work because she was helping us to do the NGS bioinformatics. But for the crypto, the students did the bioinformatics too, everything. So it's just amazing students and amazing students because I'm at the Indian Institute of Science. It continues to draw the best of the people. And if they're willing to be inspired by what ideas you give them, you can still achieve things. Thank you.